Hi guys, today we're going to have a talk about biological solutions found in nature. I'm Hashim Kale.
My name is Elena Ramos.
And I'm Kelsey Cox.
Our first topic today is BMAA and neurodegeneration. So what is BMAA? It is a neurotoxin first discovered in Guam. The acronym stands for beta-methylamino L-alanine. BMAA is produced by some types of cyanobacteria, which are found everywhere. And it is a non-proteinogenic amino acid, which means it is not produced by human and not a building block of proteins. Through biomagnification, it eventually reaches humans.
Some effects of BMAA are protein misfolding and developmental damage. BMAA substitutes itself for serine in the body. BMAA has been detected in brains of those who have had ALS-PDC and Alzheimer's disease. It also has the ability to act as an ionotropic and metabotropic glutamate receptor, where it can cause neuronal degeneration. Some current research being studied is one where a fruit fly study found that serine inhibits BMAA, and another one where the reaction of BMAA and divalent metal ions alters the rate of carbonate formation, affecting neurotoxicity.
So why do you use fruit flies as a model? Well, it's a fast, cheap solution to observe generational changes. A large sample pool allows for accurate results due to being able to grow them quickly and cheaply. The SLC7A11 genes and mechanisms are similar to humans, and in fact many genes are shared with humans, and the genome is also well mapped.
So with that said, there's hope for neurodegenerative diseases. More specifically, we can use Lserine as a possible solution. Lserine is a natural amino acid that is produced by humans, and it's vital for function. As mentioned previously, Lserine competes with BMAA during the process of making proteins, which suggests that we can use Lserine as a therapy to slow down neurodegenerative diseases. So with that said, we're going to go on to our next topic, which is the benefits of using algae. So some of the benefits for using algae in society is its possibility of using it as a biofuel. It is a better and safer alternative due to its high fatty acid composition. Another benefit is that it's a healthy food alternative, which Japan is already using algae in their diet as it provides a great source of nutrition. Algae also has the ability to remove greenhouse gases due to carbon fixation. And finally, algae is great at removing impurities in water, making it much safer to drink. So why pistachio water? For our experiment, we use pistachio water because there is an issue in the Central Valley with high levels of nitrates and phosphates found in our drinking water as well as our irrigation water. So with that said, we can use the algae to grow it in this water as well as hopefully purify the nitrates and phosphates found. And in this case, the algae strain we used was Chlorella vulgaris.
So some methods and parameters for algae growth are taking spectrophotometric measurements at 680 nanometer wavelengths and 750 nanometer wavelengths for chlorophyll and optical density. And right here pictured is a microscope view of a hemocytometer, which measures cell count following dilution. And the algal media can also be measured for uptake of nitrates and phosphates by measuring those levels.
Our recovered biomass for Chlorella vulgaris was 0.5427 grams, and we began with 1.550 liters. The nitrate concentration decreased to an undetectable level, and phosphate concentration varied, ending at 0.025 liters over the course of 17 days. Our data recorded at 680 nanometers represents chlorophyll concentration correlating to pigment concentration. There was a decrease in absorbance in our data as the concentration of nitrate decreased while the phosphate levels varied. Data recorded at 750 nanometers represents dispersion of light due to turbidity correlating to the amount of growth of the algae. Our absorbance levels showed no trend pattern with decreasing nitrate levels and varied phosphate levels. In conclusion, the proper media for growth needs to be established. Algae could potentially harness nitrates and phosphates present in water However, this experiment was in its preliminary stage and further research needs to be conducted.